Regulators on Friday approved 2 new gene therapies for sickle cell disease that doctors hope can cure the painful inherited blood disorder. The Food and Drug Administration said the one-time treatments can be used for patients 12 and older with severe forms of the disease. One, made by Vertex Pharmaceuticals and CRISPR Therapeutics, is the first approved therapy based on CRISPR, the gene editing tool that won its inventors the Nobel Prize in 2020. The other is made by Bluebird Bio and works differently. Sickle cell disease is a rare, debilitating, and life-threatening blood disorder with significant unmet need. The scientists said, "We are excited to advance the field especially for individuals whose lives have been severely disrupted by the disease." The 2 gene therapies are the first approved in the U.S. for sickle cell disease. The FDA has previously authorized 15 gene therapies for other conditions. Researchers used the CRISPR-Cas9 gene editing system to fix a mutation that caused mice to have sickle cell disease. With CRISPR, scientists correct genetic mutation that causes sickle cell disease. CRISPR technologies, how it works. Gene editing refers to the process of changing regions of cellular DNA. The most common gene editing techniques involve inactivating a gene's function, knockout, introducing or correcting a SNP mutation, or adding a reporter tag to an endogenous gene, knock-in. These changes are permanent and heritable, resulting in a newly engineered cell line. With CRISPR-Cas9 reagents, researchers can make targeted, specific edits to any gene in practically any cell type. Additionally, a wide selection of pre-made edited cell lines and expert genome engineering services are available. Sickle cell is most common among Black people, and 1 in 365 Black babies are born with the disease nationally. The disease affects hemoglobin, the protein in red blood cells that carries oxygen. A genetic mutation causes the cells to become sickle or crescent-shaped, which can block blood flow, causing excruciating pain, organ damage, stroke, and other problems. Current treatments include medications and blood transfusions. The only permanent solution is a bone marrow transplant, which must come from a closely matched donor without the disease and brings a risk of rejection. No donor is required for the gene therapies, which permanently change DNA in a patient's blood cells. The goal of the Vertex therapy, called Casgevy, is to help the body go back to producing a fetal form of hemoglobin that's present at birth. It's the adult form that's defective in people with sickle cell. CRISPR is used to knock out a gene in stem cells collected from the patient. Bluebird's treatment, called Lyfgenia, aims to add copies of a modified gene, which helps red blood cells produce anti-sickling hemoglobin that prevents or reverses misshapen cells. When patients get the treatments, stem cells are removed from their blood and sent to a lab. Before getting the altered cells back, they must undergo chemotherapy. The process requires at least 2 hospitalizations, one lasting 4 to 6 weeks. Still, many patients say they'd consider gene therapy given the seriousness of the disease.